I'm Salma Quraishi. Welcome to Neuroscientists Talk Shop, the University of Texas at San Antonio's neuroscience research podcast. It's October 22nd, 2020, and we'll be talking shop in a minute with Nicola Grissom. It's Nicola, right? I keep wanting to say Nicola. But first, I want to take a minute to make some exciting announcements. First, you may have noticed, some of you out there, that our conversations are now on our brand new UTSA Neuroscience YouTube channel. Please check us out there and subscribe. We've got lots of new content planned and underway, which brings me to item number 2. So on November 12th, we're hosting our 12th annual research symposium, which this year for the first time ever will be live streamed to our YouTube channel in its entirety, and also available to the public to join as a WebEx event. So in the past, we've only recorded just the 45-minute panel discussion for you guys and posted to our audio podcast. But this year, you'll have 2 ways to experience the whole thing from start to finish, 10 a.m. to 5 p.m. Central Time. So links to connect to either the live stream on YouTube or the WebEx event will be available shortly on our website, on the YouTube channel, and also on Twitter. So these symposia are really special events for us. For each of the last 12 years, we've highlighted the research area of one of our brilliant young faculty by having them bring in the vanguard of their field for an immersive day of research talks. So this year, Lindsey Macpherson, who is with us today. Hi, Lindsey.
Hi.
She has the honor, and she's put together a symposium called A Gut Feeling, Chemosensory Signaling from the Tongue and Gut to the Brain. So Lindsey, can you tell us what you got planned?
Sure. I'm really excited about this symposium coming up soon. I've just recently talked to our panelists, and they're also very excited to join in. So we're hoping to reach out to have a broader reach than we usually do by having this all online. And so our panelists will include myself talking a little bit about taste and gut chemosensation and connectivity using a new grasp technique that I've been developing. And we will also hear from Alan Spector, Diego Bojorquez, Yvonne de Araujo, Suzanne Appleyard, and Lindsey Shear. And they are all talking a little bit about chemosensation from the tongue and gut to the brain, and all of the fantastic and interesting new things that are coming out in this field. So I hope you all can join us.
Hey, thanks. Okay, so on to the show, most important business at hand. So today we're talking with Nicola Grissom, who is assistant professor in the Department of Psychology, and she's also part of the NeuroPRSMH Center for Computational Psychiatry at the University of Minnesota Twin Cities. Hi, Nicola.
Hello. I don't know why I waved. I mean,
why not? So broadly, Nicola's research uses mouse models and computational psychiatry approaches to understand motivation, executive function, and how diverse brains implement different cognitive strategies and goal-directed behaviors. Her work examines the impact of sex on executive function, and she has just launched a big funded program, an NIMH-funded program, to define how sex interacts with genetic factors in brain circuitry to produce male vulnerability in a mouse model of autism. So in the gallery today, we've got Matt Wanat, fellow motivation person. Hi, Matt.
Howdy.
And Lindsey, we've already heard from. Hi, Lindsey.
Hi.
And of course, MVP Charlie Wilson, to whom I wish the happiest of happy birthdays today. A lot going on today. Happy birthday. So I think I've talked way too much, and I want to allow you, Nicola, to take the floor. I think it's worth starting by saying something about how cognitive researchers have historically approached, or not approached, the matter of sex in studying behavior and decision-making, and where we are now on that front in terms of framing sex and performance through the lens of computational psychiatry in humans and in model systems, in mouse models. So can you say something about
that? For sure. The question is, can I stop saying something about it? So the question of how scientists, people have approached sex and gender, really, although obviously in animal models, we're really only going to be able to really get at sex, has sort of like 2 parts, and I'm hoping that we can sort of usher in a 3rd part. So part 1 is ignore it, right? Like, so assume that it's, you know, at best, a source of variability. This idea that Becca Shansky has been instrumental in sort of putting forward or highlighting for us, this sort of like assumption that, that, you know, female animals, girls and women and people are, you know, males, but with some extra hormones, basically, that makes them different and more variable. And so we don't really need to worry about that special case, that, you know, femaleness is a special case of being male. And we can just sort of establish, you know, the ground truth of biology, and then worry about these little details later. So it's a relatively pejorative take, but that kind of describes where we've been at, you know. And that includes like where I was at as a graduate student, you know, trying to understand one manipulation while seemingly not worrying about a different one, which would be sex. So, okay, so then flash forward to where we're at now. So there's been an undercurrent of people who have been interested in sex differences research for a while, trying to highlight that sex itself is a biological variable, and that it is not, especially not sufficient to say that, you know, that a female is a special case of a male, purely from a sort of mammalian developmental standpoint. If anything, a male is a special case of a female, given that, you know, the sort of primary developmental program of an embryo is female, and then additional masculinization steps primarily occur to cause a male to develop, which is itself oversimplified and over binary. But you know, this people highlighting that, for instance, there are more differences across males and females than just, you know, gonadal sex hormones. And people highlighting this need to understand how all these factors might cause differences that are important, has led to the sort of push for like, okay, well, let's start including, you know, sex as a biological variable, or at least including males and females in approximately equal distribution, equal number, in experiments. And this has still led to, you know, some pushback and some, you know, grumblings, including like when I go and give talks occasionally, people being like, well, now I have to double my N. You know, not necessarily, you know, you could just use the same N, but make sure that they're equally distributed across males and females. But this sort of concern that you're then going to be studying two different mechanisms at whatever level that you're trying to discover, like whatever's going on in the males and whatever's going on in the females, that you're trying to discover two things at once. And so that's, I think, the latent assumption that is operating, even if you're not controlling for hormones. And so there's a third wave that I've been thinking about, but I haven't really articulated. I've been thinking about trying to write something about this, but I am only like, I have a couple like very rough drafts in my documents, but nothing that's quite shaped up, which is that, but a third wave that I'd be hopeful for, which some of the sex differences researchers have commented on, but which is really challenging, which is that we can't treat these as binaries, right? That if we go into justifying sex as a biological variable by saying that, you know, it's probably going to be so different between males and females, that assumes that the primary state of sex differences is that there's a mean for typical males and a mean for typical females, and there's very little overlap. But in fact, for most things that we really care about in sex differences, most of what exists is overlap, and there might be a mean difference, but you cannot tell based on looking at this dependent variable, whatever it is, whether or not you're looking at a male or female. And that goes all the way from cognitive variables out to even some really fundamental biological variables that you think would be correlated. Even sometimes in humans, when it comes to what we consider typical gonadal sex hormones, there can be quite a bit of variability where you could make a significant number of mistakes if you just looked at you know, those hormone levels and decided whether or not you were dealing with male or female category. And so something that I am myself working through how to accomplish in a statistical and analytical way is to try to describe sex differences without reifying any binaries. That is, we know that none of these mechanisms are binary. We know that there's no place you can point to in the brain, especially of humans, and be like, this is always going to be different in a reliable way. This is the male version of this, and this is female version of this. And so we have to be prepared to accommodate the fact that when we're thinking about this as a variable, that it's a source of variability, but it is not necessarily a unidirectional source of variability, that there's sex is a spectrum, and it's a spectrum in multiple different dimensions, which is the math that I've been steeping in now for a few years, starting to leak through into how I think about sex differences or sex similarities or the intersection between the two. That's it in a nutshell, basically, that like I am concerned sometimes by the focus of sex difference researchers in trying to overemphasize the differences, even if you have identified a big difference in one measure. You know, I want to try to discover things about brains in general, that that can be informative to an individual human potentially down the road, regardless of, you know, their gender identity, regardless of their particular sex hormone, sex genotype, other kinds of sex variable mediated mechanisms that have uniquely combined to create this particular person, regardless of where they sit on a sex or gender spectrum. But recognizing that the biological mechanisms of sex differentiation can be one of the contributors there, you know.
So in trying to reduce this into something that has translational meaning, as well as just presents itself as a kind of a beguiling tool, we have clinical cases that have clear sexual dimorphisms and involve executive function and decision making, and that's autism. So talk to us about, because I understand that it's, I thought at some level you were going to say that, and you kind of did touch on the fact that it is a fraught thing to talk about performance as a single outcome, as a measure, as sort of an endpoint measure of something behavioral. But I think it's also interesting that you can resolve, you can sort of decompose it into process also, and kind of find a finer level of detail that then you can try to map onto something that the brain is doing in terms of process, the algorithm, the strategy, right? Yeah. So we can go either way, to autism first, or then sort of how you kind of connect some of those, whether this has made it easier to approach sex also from some of these folkloric ideas of saying, well, this study finds that, you know, women are more less exploratory, which implies, or males are more exploratory, which implies that women are less exploratory, and in mice of all things, right? So anyway, take it away. Sorry.
Right. No. Well, so there's a few things to unpack. So I do want to talk about our recent paper that's about to come out in 7 days. I can do math. It's going to come out in Neurobiology, where we looked at some male and female mice, just wild type, typical mice, as they solved a 2-arm bandit, and found that on average, there were pretty strong tendencies for female mice to solve the task in a sort of like united, similar way across females, whereas males tended to solve the task a little bit differently from themselves each and every time. It's sort of like more of a mishmash of strategies and exploration that ended up in this particular task with females learning the task more quickly and achieving higher levels of responding more rapidly.
So do you think that a lot of these things, could you maybe describe the 2-arm bandit a little bit, the task there, and if the learning you think is, you know, generalizable perhaps to, or the results you have to other sort of tasks that are looking at other elements of cognitive function?
Yes, but don't let me forget about the
piling it up on you.
No. Only because I want to, the gender bias in autism is not as clear cut as it seems at first pass, which is something important to lay down because it contextualizes the relevance of everything that we're doing here. But so the tasks that we've been setting our mice are really exciting to me for a few reasons. And what the lab has been largely working with are what's called Bandit Tasks. Bandit Tasks are kind of probabilistic learning tasks at their most fundamental. That is, there's 2 or more options. Each of them vary in their reward probability. That reward probability can change independently or together with time. The options can vary whether they're like divided by space or by image identity or something else. But what they have in common is that because of the randomization of certain aspects, so for instance, randomization of which location an image is presented on trial to trial, you can dissociate things like responding based on location from responding based on image. Because no option in a Bandit task is rewarded every time, you can look at a response to what happens after you respond to a given location or a given image, whether or not it's correct, it's more rewarding. Even you can look at the trials where it didn't happen to be rewarding that time, and likewise you can look at the rewarding responses for the option that's not likely to be as rewarding in a two-arm situation. Bandit tasks are named from the idea that a slot machine is a 1-arm bandit. So if you talk about a 2-arm bandit, you're basically comparing 2 slot machines and you want to see which one is going to pay you off more frequently. And so that sort of illustrates the problem nicely, is that if you have 2 slot machines, how are you going to decide when to give up on this one and try the other one in this really simple world? If you have 3, how do you decide which one you're switching from, how you're exploring between your 3 options? Solving these kinds of tasks is something that is of interest, not just to a mouse researcher like me, but to people working with non-human primates, people working with humans, and artificial intelligence researchers. So some of the conferences I've been able to start going to have had, that much more computational focus, and it's been a little bit mind-blowing in that way. So they're really useful for being able to dissociate situations where holding this thing constant and all the other variables around them are allowed to vary more or less randomly, which is really good for deriving clear neural signals down the road, and really good for being able to determine behavioral strategies. And what we're able to do with the mice was find that compared to, what the female mice ended up doing was they preferred to simplify. It seemed like they were simplifying the task to some extent. I don't think that this was on purpose. I don't think the female were so like, aha, I know what to do, although I can't be sure. But they would just, when they were presented with an uncertain combination of 2 images, they would just always choose whatever was displaying on one side. Every female had her own favorite side. Pretty evenly divided. We originally thought there might be a bias, but it turns out it's pretty evenly divided between whether or not they prefer right or left. And it turns out if you do that, you'll just end up sampling both of the images in that situation. And eventually what they were able to do was then switch to responding to the more rewarded image. And in fact, switch to treating the task as an image-based task. The males were doing something a lot more complicated than that. But the one thing we know for sure is that they weren't, in large part, although not exclusively, they were much less likely to use that side bias strategy to help to sort of refine their responses. And in fact, as far as we can tell, they were largely learning on some complex combination of space and image location. And so something we haven't been able to fully figure out is like if they're learning the value of, airplane picture on the left and airplane picture on the right separately, for example, as like 2 different things that they have to learn, as opposed to sort of integrating them, generalizing airplane image, which it seems like is something the females were doing more quickly because of choosing to not explore in space as much. So in terms of what that means for sex differences, it's really striking to me that there are some males that were able to use some version of the female strategy, but as far as we could tell, the females, there didn't seem to be a lot of females that were doing as much of what the males were doing or had that same level of variability. But there's certainly room for that to be true. But more than anything, it just really revealed to us that this really simple task, 2 images that can just sort of vary in location, and one pays off 80% of the time, and the other one pays off 20% at a time, that alone is enough to totally explode the realm of possible response options. And that explosion of possible response options allowed us to see this relatively robust distinction in strategies that happen to separate in these mice according to sex in large part. Does that mean that sex is the only thing or the most important thing that might explode those strategies? Probably, maybe not. Does that mean that it would work the same way in humans? Well, they're probably not going to assume that this is a spatial task early on, but it might give us important information about how humans are integrating information and choosing to obey different kinds of rules. It might give us the tools that we had to develop to describe the strategies that the mice were doing computationally. It might give us the tools to understand the change in strategies that might best describe, somebody with depression or with some subtype of depression, for example. Or one of the reasons why I got really interested in this might help us understand why there are differences in exploration seen on average in people who are on the autism spectrum compared to neurotypical people. And that is one of the robust Bandit task findings that exists, is that people who are on the autism spectrum tend to explore a lot in these kinds of decision-making tasks. So in trying to decide between these two arms, they will explore for a lot longer than neurotypical people will. And there might be situations in which that works out, and there might be situations where that's a lot more challenging, but I think it can reveal, as came up in the talk earlier, and was really nicely put by Charlie, it can reveal a sort of algorithm that might be really foundational to a lot of the different challenges that people on the autism spectrum face. And allow us to sort of get at it mechanistically in mice without assuming things about a mouse's social world, for example, or assuming things about their preferred repetitive behaviors. So that has led us to that interest. But with the caveat, so bring it full circle. In trying to relate this work to autism, the same kind of challenge, it seems like a similar challenge to me as trying to talk about sex differences. So something that I didn't get to say early on, but I meant to emphasize when it comes to sex differences, is that there is if you look through the literature, the older decision-making literature, or even some of the newer stuff, the assumption is that if you have a difference in decision-making behavior, it's because one group is better than the other, right? Like, so what do you do if it's a sex difference in behavior? Who's better? How do you decide that? It's arguable that neither is better, and that's not the right question to ask. Because there might be situations where one particular response strategy is going to lead to, maybe better outcomes. There might be other situations or other kinds of outcomes that you want to prioritize, and that's going to lead to better outcomes for that particular set of strategies. And so my thinking about sex differences and wanting to be very careful that we don't assume that we as scientists know the right answer just because we designed the task a certain way, also feeds into how I think about autism, wanting to make sure that we don't assume that we know everything about what an animal should prioritize in this task. and how they should solve it, given that they are telling us what they think of a strange situation. And that, I think, gives me some empathy for trying to, or it leads into some empathy for trying to think about how we interpret decision-making differences across humans, across human gender, or across human psychiatric conditions, to not assume that because we identify a difference, that difference is necessarily a problem. It might be a challenge. It might be part of a deficit or something that we want to address. But the onus is on us to make sure that we're not pathologizing things, getting ahead of ourselves, because otherwise you end up in a situation where, for instance, Iowa gambling task, something I do not like. I do not like the Iowa gambling task, but there's a lot of Iowa gambling task papers you can find out there, or there's sufficient, where, you know, women don't choose the particular deck of cards that the task designers decided is the most optimal. And so they're like, well, obviously, you know, there's a problem with decision making, as opposed to looking at the kinds of sequences that are preferred, and or the kinds of decks that are preferred, and realizing that they're optimizing for a different solution, which might be to avoid frequent punishment. As opposed
I'm always
to
worried that I'm going to choose the wrong one of those gambling tasks. I don't think I could actually figure it out, and I don't know. I'm afraid of actually testing myself to see if I would pick the optimal or non-optimal one. And does that mean anything? I think you're right with that.
Oh,
But,
this is this is the difference between us, is that when I do those tests, I'm like, and I don't choose what's up. I'm like, this is dumb. Like, who decided that this is the optimal choice?
Right, it's optimal for me. Because I do feel like I feel the loss very acutely, and so like to lose something, I hate that. So I think for me, I would avoid those punishments much more than gain. I would prefer to gain a little bit less and avoid the bigger punishments.
Right. And so is that economically?
Yeah.
No, but I don't think our brains evolved in economic systems. We evolved in like foraging like environments where you know, avoiding losses of any kind to maintain a steady state has a particular value. If there's a
have you been a subject in one of these tests or something? How do you know about this?
Oh no, just from talks. Like I've heard people give talks about these things, and I'm always a little bit worried that I would always pick the wrong one. I would never figure out the optimal.
When I went to Iowa to gamble, I always went to Vegas.
I love going to Vegas. I find it extremely exciting, but I as soon as I start losing, I'm out. Like
the weirdest thing though is we always assume the simplest and most optimal strategy. Gambling is like famously never the simplest. There's always this top down, you know, rubbing rabbits' feet, and I mean, it's so fraught. You know, so why do we assume that the simplest strategies are going to be what is happening at the implementation level, or I guess we test for that. We have to test for it.
No,
how do we know that?
Oh no, you highlight the problem perfectly, which is that we design really simple tasks in the laboratory, and then we set them to the animal. The animal doesn't know what the hell is going on, or if it's a person, like, you know, they don't know what the rule is, and so there's nothing stopping them from assuming that the rule is more complicated. And in fact, it might be more complicated in a real life situation. So like, it's just like, of course the simple strategy isn't going to always be the clearest one, because when you're in a real life gambling situation, or the complex environment of the real world, that's not how it works. You know, assuming that that an animal that's not following that super simple rule is objectively non-optimal by an economic rule, but assuming that the economic rule is the most important one is, I think, not a great assumption. I'll put it that way.
What is the Iowa gambling task?
It's a task. It's a card-based task. Like, it comes from that sort of neuropsych tradition, as like card sort, other kinds of Midwestern card tasks, basically, where you're trying to understand the influences of losses and gains, basically, on decisions. And so it's designed to sort of, you know, identify big differences in, like, let's say neuropsychiatric or neuropsych populations, so like population or neuropsychiatric compared to control. That's three. 4 decks of cards. Each one has some combination of rewards and punishments, where the rewards, or excuse me, the punishments in particular, are conflated into frequency and severity. So that is, there's frequent small punishments and there's large infrequent punishments, but there's never like infrequent small punishments isn't really like as much of an option, or vice versa. Although frequent large punishments are not. And so the problem I have with the task is that if you have somebody who's avoiding, the optimal deck has a bunch of frequent small punishments in it. So you just have to be constantly be like, well, you know, gain 60 but lose 30 on this card. Well, I gained 30. And you just have to constantly, ignoring the fact that you're losing every time you're and focused on the overall economic gain of that option. Whereas one of the decks that is in particular, it seems like it is this deck that has like really infrequent big losses. So you think you're doing great, and then it's like, boom, a big loss. And so in the long run, 100 cards later, you'd be in a bad shape if you chose that deck. But who knows? They don't know that. They're picking these decks in real time. So it seems like it's a great deck, and it doesn't have a bunch of these little nonsense punishers that are going along the way in the objectively good deck. So anyway, I ranted about it in a review paper that came out a couple years ago. Me ranting about behavioral tasks. What gets me?
Could you perhaps rant on another topic? So I'm curious, how can we study, and by we I mean you and the scientists who are studying autism using preclinical models? I mean, I know there's a number of models out there, and which one do you use? Why? And I mean, what are the challenges? I mean, that we can have with having a good model? I mean, yes, just go.
So by model, do you mean the mice or the behavior, or both? Or?
Well, yeah, I think both are. You know, how can we look at something that is challenging? I think to be able to port down to mice. I think in a way. I mean, yes, there's a lot of social components that you find in autism spectrum disorder. That's one of the major things, but we might not necessarily have a full repertoire, or we can observe the full repertoire of social behaviors in rodents. And yes, I mean, what model do you use? What behaviors are appropriate to look at? I mean, it's challenging and exciting at the same point to, be at the forefront, to be, exploring these ideas.
It is both challenging and exciting. I have a particular viewpoint on this, although it's one that I think is shared by a variety of people talking, casually, at conferences or things like that. That doesn't always come out in the publication record, although it's coming out more and more. Which is
that's why we're here, for you to discuss these things off yet on the record.
On the record, but without having to write a whole paper about it. So a lot of the behavioral tasks that people sort of think of with regards to autism are not, I mean, in mice, and trying to model autism relevant behavior in mice have really big construct validity and sort of computational challenges that make it really sort of not great. And by not great, I mean both questionable about like what they're going to actually tell us about autism per se, and when one of the big autism meetings that happens in the springtime happens, they tweet a lot about it. And you can read the tweets from people who are on the spectrum being like, what is this what does this behavior in this mouse have to do with my life? Zero. And the thing is, they're not wrong. They really aren't. That's the truth, in my personal opinion. So one of the big ones that I'll occasionally rant about is at the behavioral test that's used is marble bearing. And the reason I rant about it is because it's a really short, simple task. That is, you're putting a mouse in a clean cage with some bedding and some marbles, and you're measuring what they bury. But it is not at all clear why they're doing it. What is the correct number of marbles to bury? What does this have to do? How does this relate to autism? And so you'd be like, well, it has to do with repetitive behavior because they're burying them maybe because they're anxious. Except mice also bury food, and as far as we can tell, by the way, it doesn't look like they're intentionally burying the marbles at all. They're just digging and then the marbles happen to fall in. That's true across multiple labs. It's a behavior sensitive to benzodiazepines, but so are a lot of things, like they're, just moving around less. As far as we can tell, is it validated maybe as a kind of test of anxiety in the same way that the forced swim test was validated as a pharmacological test for antidepressants without having anything to do with depression-like behavior. And yet you sort of conflate the two, like because this behavior leads to a change and is changed by this drug, therefore the behavior that is being changed must have to do with the psychological construct we sure want this drug to treat. So that's not great. The lack of clarity with marble bearing combined with the fact that it's just fall off a log easy to do means that it's right for people who are doing really sophisticated molecular work to be like, and then the mouse that I've characterized, this neuronal subpopulation 6 ways from Sunday had a deficit in marble bearing and I've solved autism, So that's not great. The construct isn't clear. The construct that is testing isn't clear at all on multiple levels. What is the mouse doing it? What construct does it relate to? It's a truly messy task that I'm not doing justice here. But I'll do better. I'll do better justice in another tweet thread one of these days. Okay. So we say, well, no, that's because autism is a social disorder. It's primarily characterized by these social interaction challenges, and so you need to be measuring social behavior. This is where it gets a little more fraught, but I'm backed up by a huge file drawer effect. So one of the really classic tasks that people have said, oh, okay, well, we need to measure social behavior in autism, mice carrying autism-associated genetic variants. We're going to do the 3-chamber social interaction task. Except that, Jackie Crawley, who developed the task, has said that you're not supposed to compare a difference in the amount that they prefer. So in this task, there's a mouse under one cup and an inanimate object in another cup. And then you're looking at what your test mouse is doing to investigate the inanimate object versus not. And if both your control and your putative autism genotype prefer to investigate the social mouse at the cup over the inanimate object, even if there's a difference in the total number of seconds, there's a social preference. And that's what Jackie always said. So that's the first problem, is that it was interpreted as like, well, if there's a 3-second difference between how much they're sniffing the social cup, that's a social interaction deficit, which is not true. You can't really say that. But more importantly, why would a male mouse investigate another male mouse trapped under a cup? It's not usually to be best friends. And it sort of underscores a messy secret with that task, which is that if you ask 3 different labs who that animal underneath the cup is, you'll get 3 different answers in terms of, is it a cage mate? Is it a conspecific of the same age? Is it a different strain? Is that strain gonadectomized or not? And I think can lead to a lot of differences there. But more to the point, when you look at the actual social behavior that people on the spectrum show, it's it's not that they don't like to have friends or that they don't necessarily want to interact with people, but they don't always want to interact with people in sort of the neurotypical ways. And they might have real challenges, you know, sort of both understanding subtleties of communication and wanting to communicate in the ways that neurotypical people would prefer them to. You know, like verbalizing or with, you know, not with combined with like complex nonverbal communication, like differences in tone and stuff like that. So, you know, this mouse choosing to investigate another mouse was, you know, originally it was sort of a big hit. It's like, oh, it's a way to measure social behavior. Except then when we finally got well-validated genetic models, so like mice carrying genotypes associated with autism, most of them don't show deficits in that task. And that's published as a news article, not as a scientific publication, but the challenges with that came out in like Spectrum like a couple years ago. With a really depressing title. It's like, you know, can we ever study autism in mice? No, probably not, something like that. So, all right, so if we're going to try to understand anything about autism in mice, and we're going to attempt to do it with like a modicum of hope for translation and sophistication and you know, respect for the lived experience of people who are on the spectrum, we have to try to meet them where they're at, you know. And what do people on the autism spectrum report? Is their problems anxiety and executive function. They are anxious, off the walls, and that might have to do with, you know, like some of the predictive impairment stuff I was talking about during my talk. That is, if the world seems less predictable, then yeah, it's going to be super anxiety provoking. And it makes it also much harder to plan and focus. And so that's really informed my research through my postdoc, was to try to look at executive function in particular. And ultimately, it's what led me to thinking about decision-making and potential sex differences in strategies, is that, you know, if there's a role for biological sex mechanisms beyond hormones into, you know, genetic sex, into the numerous mechanisms of early life by which all sexual differentiation is having an influence. If there's a role on decision-making as an aspect of executive function, then that might be something that we can really mechanistically access. It does not mean that's the only way anybody's ever gonna be able to get at this in mice, but it does at least help me sleep at night that I'm not trying to, you know, equate the social world of a mouse to the social world of a person. And like I said, that doesn't mean that that wouldn't be possible to do, but for my very discrete goal, which is, you know, cross-species, computationally validated approaches to trying to get at the same cognitive and computational constructs, algorithmic constructs across species, I need something that I can sort of point to and say like, okay, we at least, ask the same question. We ask them, you know, what's the next decision you're going to make after this history of previous decisions and outcomes. It doesn't mean that humans and animals are going to solve that the same way. In fact, they almost certainly aren't, but it might help us to understand the mechanisms in the brain that would lead to a particular kind of decision after a particular kind of history. And that might translate, and that might also show room for sex, we know, and room for autism, we think, based on human and non-human data. Big gamble, but really long soliloquy, but there you go.
Yeah, that was excellent.
That's such a great note to end on, but I do want to ask you one more thing if you're not too exhausted. We'll limp through it. Let's see. So I'm just wondering if the idea is, so I think I've heard David Reddish say that, or maybe he says it about the DSM, not about autism, but that it's a a set of symptoms, not a syndrome. You've heard it too, maybe.
I do get to talk to
Yeah. Yeah. So I wonder and I know at some level that, you know, you've defined your set, your scale and your set of tools, and you're sort of thinking of it in a particular way. But do you imagine that this is just a bunch of different insults that all sort of land on maybe different architectures, different hormones, different proteins being expressed? Because, you know, a lot of this stuff is, you know, we're talking about genes and genetic loci, but ultimately genes code proteins. And, you know, I just don't know. I don't know how you get from proteins to like decision making, right? So is there just this idea that irrespective of whatever, you know, the biological substrate, the architecture, whatever you want to call it is, that there's some solution in the implementation of whatever's happening that you can manipulate pharmacologically? Because that's the other thing, is that psychiatry is ultimately, I mean, what are the tools of psychiatry? They're drugs and maybe ECT and maybe some behavioral, I guess, shaping, some sort of learning type thing. But, or enrichment, I guess. I don't know what the clinical interventions for autism are. So I maybe you can even tell us. So what is the sort of black box that separates those things in terms of, do you think that it's somebody else's job to get at the architectures? Are you interested in the architectures, or you really think that this is just happening at a level that's beyond circuits, for example, or neurons, or, you know, transporter protein, you know, that level of protein?
So, okay. Yeah. So no, it's a great question. People originally thought, you know, when we started to get genomes, that the solution to all of our psychiatric diseases were going to be genetic. That we were going to find genes, those genes were all going to have a common mechanism. And then we've done it, and at least for, you know, where we're at now for autism, you know, there's a lot of different genetic factors, and they don't all have a common mechanism. There may be, you know, 3 to 4 mechanistic themes, but they basically go everywhere from, well, the synapse, but also the nucleus, which is not particularly helpful. So, so that, where does that leave you? Well, so given that we have this sort of behavioral thread that we can draw, behavioral cognitive thread that we can draw across species, through like decision making, let's say, how do you get from these genotypes to that risk, or, you know, to differences in cognition? So I am interested in that. And so one thing you might think is, well, there's going to be a circuit level convergence, which is something I'm sort of currently writing grants about now. And not that I think that it's going to be that simple, but because that's the sort of parsimonious place to start, right? Is that there could be a circuit level convergence if you test multiple different genotypes. Either at the sort of macro circuit, like within a region, so like within the striatum, you know, D1, D2, or for the other grant that I have within the prefrontal cortex, the, you know, synaptic, the activity correlation, coactivation of multiple neurons within the PFC when animals are making decisions, blah, blah, blah. But you know, there's really interesting ways that you could think about neurodevelopmental mechanisms as being able to play a role in how these decision-making systems function. And so just as like a sort of like teaser note, like some of this stuff is just coming out now, but I was just discussing with some colleagues earlier this week, this really interesting preprint from Xiaojing Wang's group at NYU, where, you know, the paper is mostly modeling, but they start by saying, well, you know, let's look at the expression of D1 receptor across cortex in primate. And what they find is that more or less, you know, cortex, visual cortex, frontal cortex, parietal cortex, you know, you'd think it's all the same, but in fact, there's a really sort of overt anterior posterior gradient in the density of D1 receptors in cortex as a function of of sort of like lowest in V1 and sort of hindbrain up through highest in frontal and some parietal regions. And that lends itself directly to a neurodevelopmental mechanism, because then you say, well, we know a lot just from developmental biology about how things like anterior posterior gradients are going to get laid down. Then it's no longer a mystery. And what they were able to show in that paper, or what they did, is then they modeled a brain that had a really simple, two pyramidal neuron cortical region across all these regions, and found that the ability for this complex biophysical model to maintain working memory, is what they had the model work on, was dependent on a sort of optimal dopamine release, or optimal dopamine availability. And they were able to replicate an inverted U function for dopamine release in this model as a function of just adding the different sensitivity of different regions front to back to dopamine. So that's really potentially exciting, because they didn't talk about this in the paper. They're modelers, they're not developmental biologists, but it does suggest, okay, so this one thing that might be happening is you need to regulate how pyramidal neurons are sensitive to dopamine. You might also need to then use those same mechanisms to regulate, some of those same molecular mechanisms might be involved in the striatum, regulating how they're expressing their receptors. One thing we know in one of the mouse models I really like to work with is that there seems to be potentially, at least in early life, an increase in double labeled D1 and D2 neurons. Not a huge number, but way more than you see in wild type. We don't know if it persists into adulthood, but we have some evidence that is not inconsistent with that idea, I guess. So you can begin to draw a line from like, okay, you have a molecular problem. It's going to cause some difference in the development of these circuits. And the common circuit mechanism might be not that this brain area always bad or always goes down or isn't working right, but that the really complex interplay between these regions is being tweaked at a molecular level, but at a molecular level that's sensitive to where you are in the brain by these neurodevelopmental mechanisms. And then that leads to a circuit that then we can model and say, like, actually, this model doesn't quite solve the task as normally. So that's a big job, which is partially why we have the P50, is because there's pieces of that all, and that one's particularly focused on psychosis, but there's some shared mechanisms there, including shared genetic models. Most genetic models of autism could also be argued as genetic models of psychosis on some level, which is a podcast for another time.
Yeah, that's counterintuitive. Thank you for staying over time with us. I realize we've gone a few minutes over, and this is really just lots of fun. I feel like we could just keep throwing stuff at you, but we'll give you some relief. But thank you for joining us, Nicola Grissom. This has been Neuroscientists Talk Shop. Thank you, everyone.
Thank you very much. Have a wonderful evening.
Yeah, you too.